All right, so we chatted in school about structure of DNA a little bit. We did talk about its discovery. Main part of it, though, we got to chat about that we need to add to is a little bit on the numbering system, and then we got to look at how it replicates, and that's what the video tonight's going to go through. So DNA itself is a deoxyribose sugar. That's the base part of it, and then it's got a phosphate group as well as one of 4 nitrogen bases. And we've all seen probably at some point this picture on the right hand side, right, where we've got this double helix structure, maybe even seen something like this one on the left. Again, still the double helix structure. Important thing here, we are not trying to choose a color. Important thing here, though, to look for is the fact that if you look, every one of these A's is bonded to a T. Every G is bonded to a C. Now, they're not actually this pointy shape or the rounded shape, but it's that. The idea is that G's match with C's and A's match with T's. And you do need to know what those 4 chemicals stand for. So just to take a look at those real quick, we have A is adenine, G is guanine. Chemically, they are classified as a purine. T for thymine and C for cytosine. These are pyrimidines chemically. The odd time they do ask a question about that, it's annoying, but that's the way it goes. Something that you basically just need to memorize. In terms of the numbering system, so the A's and the T's and the C's and the G's, if we were to look at each of the corners of these hexagons, or the hexagon and then the pentagon, each of those corners is a carbon with enough hydrogens attached to it. And then if we look here at, and they would be numbered 1, 2, 3, 4, 5, 6, or 1, 2, 3, 4, 5, 6, 7, 8, 9, or depending on how many carbons there are. But when we get to the deoxyribose sugar itself, the sugar in this corner is the 1 prime. This corner is 2 prime, 3 prime. Notice that 3 prime, it's supposed to be like a prime or an apostrophe sign. My mouse is not cooperating. 4 prime is this one here. And then this last C to H that it's got here, this is our 5 prime carbon. So if you look, the 5 prime carbon is attached to the phosphate group, so is the 3 prime carbon. It's just that the 3 prime carbon is attached to the phosphate group from the one below. And so if you want to think about it, the O is like a little arrow and it's pointing up. And or in this case, it's pointing up anyways, as in we're going from 1 over here up to 5. All right. And that's sometimes not a bad way to try to remember how those are numbered off. If we look on this side here, again, this would be 1 prime. Here's 2 prime, 3 prime, 4 prime, and that 3 had an issue. That didn't make that issue a whole lot better. And then down here is our 5 prime carbon. All right, so again, it's almost like we're going from 1 over here around and we're counting that way. All right, now coming back to this bit here, nitrogen bases are attached to that 1 prime carbon. And if we talk about adenine and thymine, guanine and cytosine, in any species, the proportion of A is equal to T, which makes sense because all the adenines are bonded to a thymine. Proportion of G is equal to C. Again, makes sense because guanine is equal to cytosine. And essentially, A plus G is equal to T plus C. What that really means is that if I give you any one of those four, you can figure out the rest. So let's say I told you that in a certain species, 21% is thymine of the DNA. So what we know, come on, mouse is being obstinate here. 21% is thymine. There we go. So thymine is 21%. All right, so using Chargaff's rules, we know that A plus G is equal to T plus C. A plus G equals T plus C. We know T is 21%. We also know that from down here, A is equal to T. So 21 plus G equals 21 plus C. And the last little bit that I haven't told you yet, but it's not explicitly stated anyways, is that, of course, A plus T plus G plus C is all equal to 100. And if we know G and C are the same, well, what do we know here? We could look at it and do some algebra here, right? But the big thing that we also can know is that, well, if A plus T, that's 42, that means G, this is the easier way to do it, I'll be honest with you. That means 42 plus G plus C equals 100. Which means G plus C is equal to 100 minus 42, which means G plus C is equal to 58. Come on. And we also know G and C are the same, which means G plus G is equal to 58 essentially, or I can take 58 and divide it by 2, because I know G and C are the same, and I realize that, oh, those are 29 each. So given that I started with this one, T is 21, I know that A is 21. Oh, 21%. A is 21% as well. Once I know those two things, I know, oh, okay, A plus T is 42. I'm left with G plus C having to make up the rest of it. They're both the same, which means G is going to have to be, in this case, like we just said, half of 58, which is 29. And C has also got to be 29. And again, it hopefully should make sense that if G has to be bonded to C, it means there's got to be the same amount of each of them. If A has to be bonded to T, there's got to be the same amount of each of them. All right, and so that comes from this guy, Chargaff's rules. Moving along, we chatted about Watson and Crick and Rosalie Franklin. I showed you that picture. You can look it up too on Google, of Watson and Crick building this model of DNA in 1953. Now, what I didn't really stress maybe enough is when they built that, it was like all the bond angles were correct. The spacing of the atoms was like perfect. This was not a little Mickey Mouse. Everything was to scale. It was really well done. Come on. There we go. Oops. Which takes us on to, what does DNA actually look like? Well, as we looked on the diagram, it has two strands of nucleotides. Okay, those are those A's, T's, G's, and C's. They're complementary, and when they form these complementary base pairs, we call this bond between A and T a complementary base pair. They're hydrogen bonds. I've never seen this specific information on a test, but if you want to know why does A only bond to T? A and T form 2 hydrogen bonds. G and C form 3 hydrogen bonds. And just the location of the elements required for these hydrogen bonding lines up with the A and the T, also with the C and the G. And so an A does not line up properly with a G. And that's why we don't end up with them. Now, when we talk about the DNA strand itself, we can talk about the 5' end and the 3' end. And if we look back at our picture of DNA that we drew all over, come back here, there we are. This is our 5' end up, 3' end going down. So when we talk about a strand running from 3' to 5', it's running from 3' end at the bottom. So here's our 3' carbon running up to 5'. So this strand over here, runs from 3' to 5'. This strand over here runs, again, going the same direction, so from the bottom up, from 5' towards the 3' end. Right? And that's, so a little bit of kind of naming stuff and a convention on how we look at these things. And the way we would give the information of which nitrogen bases are there is just using those letters. And so if we say from the 5' end, we have a T, C, G, A, G, G, C, T, T. Reading the complementary strand, well, T goes with A, C goes with G, G goes with C, A goes with T, C would bond with that G, C would bond with that G, G would bond with that C, A would bond with a T, A would bond with a T, and so on. So if we know one of them, we can deduce what the other one is. We say the strands, if you want, mathematically, they are anti-parallel, right? So they're parallel, but they run in opposite directions. All right, so at one end, we have the 3' carbon and a hydroxyl group with a deoxyribose sugar. Other end is a 5' carbon and a phosphate group. Now, DNA is replicated during S of interphase. And DNA replication, by definition, is where a cell makes a copy of its own DNA. Now, this replication is what we call semi-conservative. So as opposed to being fully conservative, or sorry, I should say, or being non-conservative, would be more like we take the DNA and make a photocopy of it, right? You end up with your original and you end up with a copy. DNA replication is not like that. Okay, it's not like we take the DNA to the photocopier and we just make a, zap, 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 and there's our copy of it. And this was how Watson and Crick hypothesized that it should happen, and two guys, Maselson and Stahl, experimentally confirmed that. So they're pretty clever, actually. Again, this is using a little bit of isotopes. It was the interesting bits and pieces that they did with this. So they grew E. coli cells in a heavy nitrogen, so nitrogen 15 source. Now, they extracted the DNA and they centrifuged it. They ended up with a low band in their centrifuge or their test tube. Okay. They took the culture out of the heavy nitrogen source and they placed it in a light nitrogen source or a nitrogen 14 source. They allowed one cell division cycle to occur. They extracted the DNA, they centrifuged it. Instead of a low band, now they've got a medium band. Allowed one more cell division. They extracted the DNA, they centrifuged it. They saw a high band and a medium band in their test tubes. Okay. Now, this is what they hypothesized was occurring. All right, so we've got the colors actually mean something here. In this case, the pink is DNA that was grown in a heavy nitrogen source. And if we look at that very first centrifuge experiment, both strands are made with heavy nitrogen. And so we end up with this low band on the test tube. All right, after one cell division in the light nitrogen source. Again, this is assuming it's semi-conservative. Now, these two pink strands, they split apart. So here's one of the pink strands, which I'm trying to draw on. There we go. Now here's the pink strand here. Here's the other pink strand. So they got spread apart, separated, and then a new strand of DNA forms from the light nitrogen source, and that's shown in blue. So what we've got right now is this medium heavy band of DNA. All right, now if each of these separates, now we're going to get, we still have our heavy stuff, separated, spread apart, and copied. Then we've got over here, we've got our light nitrogen. So we've taken this blue one here, is the template strand here. We make a new one using, again, the light nitrogen source. Heavy strand here, we get the light nitrogen as well. And guess what we end up with? We end up with a medium band. Medium band shows us we have one heavy strand and one light strand. And we have this high band showing that we have the two light strands. All right, so kind of a clever experiment using a centrifuge to show, we can't weigh the difference between nitrogen and 14 and 15 on a regular lab scale, but they're going to travel different amounts because of the different force of gravity acting on them. All right. So each individual hydrogen bond is not super strong. But if we look at the sum total of all of them, there's a lot of actual hydrogen bonding between purines and pyrimidines in that DNA molecule. And so there's an enzyme required to do this. That enzyme is called DNA helicase. You need to know this enzyme. As this separation occurs, so it unwinds the helix, it breaks those hydrogen bonds, and then there are proteins that bind to the DNA strands to actually hold them apart. All right, so it's not like it's just, briefly pulling them apart and they snap back together. There's proteins that are holding them apart. That's step one, is separation. And there's more on this diagram than we've studied so far. However, this DNA helicase, there it is, prying the two strands apart, unwinding them, in fact, as well. So you're losing, for temporarily anyways, this helical structure. What you can see here is that semi-conservative bit as well. Original strands are shown in yellow, new strands shown in green. Let's talk about this primer and Okazaki fragments and lagging strand and leading strand and so on. So building it. Once that DNA molecule is separated, the new strands are built simply by complementary base pairing by another enzyme, DNA polymerase 3. All right, you need to know this enzyme. It always starts at the 3' end of the old strand and works towards the 5' end of the old strand. Okay, so it's always going to move from 3' to 5'. So on one of the two strands, the replication starts on the 3' end of the old strand when an RNA primer, sometimes called a primase, attaches to the template strand, and that tells DNA polymerase 3 where to start. This strand is called the leading strand. So if we go back here, here's our RNA primer. Okay, so this isn't DNA, it's an RNA bit. It starts there. It tells DNA polymerase, this is actually DNA polymerase 3, where to start. So it started here. It's moving towards DNA helicase. Doesn't work, and you can see that already in this picture. It doesn't work on the other strand, on the 5' strand. Because again, DNA polymerase 3 is only going to work from 3' to 5'. So on the leading strand, perfect. It's just going to be able to chase behind DNA helicase. But on the other strand, it needs to start and move backwards. And if it separated the whole way out and then started, that's not going to work so well. All right, and so on the lagging strand, periodically the RNA primer attaches. DNA polymerase 3 works from the primer back towards the 5' end. Right? So a number of RNA primers are inserted along the second strand. It's called the lagging strand. And the fragmented pieces of DNA that are replicated are called Okazaki fragments, named after the researcher who discovered them. RNA primers then, both the lagging and leading strand, although on the leading strand there's only one of them. Those RNA primers are cut out of the lagging strand and replaced with nucleotides by DNA polymerase 1. Named in order of when they were discovered, DNA polymerase 1 was the first one. DNA polymerase 3 was the third one. We don't have to worry about DNA polymerase 2. And another enzyme you need to know, so this is an enzyme heavy section, is DNA ligase. It comes along after DNA polymerase 1, and it links all the backbone together. So DNA polymerase 3 works from an RNA primer. What is an RNA primer? Well, an RNA primer is a section of RNA that bonds to the DNA, and it tells DNA polymerase 3 where to start. DNA polymerase 3 always works from 3' to 5'. So on the leading strand, RNA primer attaches once. DNA polymerase 3 can chase along after the DNA helicase. The other strand, occasionally as enough of the strand is opened up, DNA polymerase 3 attaches to, the RNA primer attaches to it, and then DNA polymerase 3 works backwards towards the break, or backwards towards the RNA primer. Those RNA primers are then cut out of the lagging strand and leading strand, and they get replaced with nucleotides by another enzyme called DNA polymerase 1. Now, that leaves notches in the chain. The notches are really the joins between the hydroxyl group and the phosphate group. A DNA ligase comes along, links all of the backbone together. Come on. Slow computer here. Come on. Sorry, guys. All right. Another picture, a little bit simpler. My recommendation would be make yourself a picture of this. This is actually a decent quality one. Very easy to draw, and that's what I like about it. So DNA helicase is this wedge unzipping the DNA. Here's our helix destabilizing proteins. Here's DNA polymerase, again, DNA polymerase. 1, 2, 3. These little gray hashes are RNA primer. Here's our continuous strand. You can see our Okazaki fragments. So my recommendation, draw it out for yourself, showing what's happening with RNA primer or RNA primase. DNA polymerase 3, DNA polymerase 1, and then DNA ligase. Okay, here's yet another picture. I kind of like this one. It defines all the things you need to know. Take a look at it, and you can use that one as a nice overview of the whole process. Finally, DNA polymerase 1 and 3 act as proofreaders, and they will check the new strands. Occasional mistakes do occur, where a T is brought in instead of a C. A G is brought in instead of an A. Those kind of things. In that case, the DNA polymerases backtrack to the mistake, cut it out, replace it, and continue. All of those repairs have to be made prior to further replications, and that's going to prevent errors from being passed on in further replications. Which is going to lead us to another video coming up on gene expression. Thanks.